Welcome back. I am so pumped you're here today because we have an absolutely mind-blowing journey ahead of us. We're going to take a high-energy, whiteboard-style crash course through the grand unified story of life. We are starting from the absolute smallest microscopic blueprints that literally make you who you are and resuming all the way out to the macroscopic level. We're talking massive populations and the entire biosphere. It's a lot to cover, so let's dive right in. Here's our roadmap for today. We're making 4 massive leaps in scale. First up, molecules and genetics. Second, homeostasis. Third, animal behavior. And finally, population ecology. Let's get to it. All right, kicking things off with part 1, molecules, enzymes, and genetics. Basically, the blueprint of life. To really grasp biology, we have to start with a totally foundational question. What is the central dogma of molecular biology? I know, it sounds incredibly intense, but honestly, it's just the ultimate biological recipe. It's the core rule explaining exactly how genetic information stored inside a cell actually becomes a living, breathing, physical reality. Let's trace how this information flows. Think of it as a 3-step process. Step 1. DNA stores your genetic info, replicating itself during the S phase of the cell cycle. And hey, fun fact, if you uncoiled all the DNA in your body, it would stretch to Pluto and back. Crazy, right? Anyway, step 2 is transcription. An enzyme called RNA polymerase steps in and synthesizes a copy of that DNA into messenger RNA, or mRNA. Finally, step 3 is translation. That mRNA travels over to the ribosome to facilitate protein synthesis. Biology is ridiculously precise about this recipe, and it's happening inside you millions of times a second. Translation always kicks off with a specific start codon, AUG, and it perfectly wraps up at stop codons like UAG, UAA, or UGA. And just like that, you are built protein by protein. But here's the thing. Proteins don't just sit there acting as building blocks. Some actually act as enzymes to catalyze metabolic reactions. Now, there are a couple ways to look at how these enzymes bind. First is the lock-and-key model. This idea suggests that the enzyme's active site and the substrate have rigidly specific, complementary geometric shapes. Basically, they just fit perfectly right from the start. But then we have the induced fit model. This one is way more dynamic. It suggests that when the substrate binds, the amino acid side chains in the active site actually change their position. The enzyme literally shapeshifts to get a perfect, flexible fit. Beyond just enzymes, your DNA is what dictates your traits through Mendelian inheritance patterns. And these traits can show up in a few distinct ways. We've got complete dominance, where one trait just totally masks another. But we also see incomplete dominance, which creates a blended, sort of intermediate phenotype in heterozygotes. Then there's codominance, where both alleles are expressed equally at the exact same time. And finally, we have sex-linked inheritance, which explains how certain genetic disorders get passed down, like hemophilia, which is a condition where the blood just can't clot normally. Moving right along to section 2, homeostasis and human systems. Let's look at the human machine. The absolute crucial point of homeostasis is surviving environmental extremes, like freezing cold or blistering heat. Just think about how warm-blooded animals manage thermoregulation. When you're freezing, your body uses vasoconstriction. It narrows your blood vessels to keep that heat trapped inside. But when you're overheating, the exact opposite happens. Your body triggers vasodilation, widening those blood vessels to boost blood flow and rapidly dump that excess heat. It is just a brilliant, completely automatic balancing act. And we see this exact same balancing act going on with your blood sugar. You can imagine insulin and glucagon as these biological counterweights. When your blood sugar runs too high, insulin promotes the conversion of glucose into glycogen so it can be stored safely away in your liver. On the flip side, when your blood sugar crashes, glucagon swoops right in to convert that stored glycogen back into usable glucose. Working together, they keep the human engine running perfectly smoothly. Okay, but what good is a running engine if it can't actually move? Think of muscle contraction as this rapid-fire chain reaction. Step 1, a nerve impulse, or action potential, triggers the response. And get this, these signals can travel at speeds up to 270 miles per hour. Step 2, that action potential forces the sarcoplasmic reticulum in your cells to dump calcium. Step 3, that released calcium allows your 2 main muscle filaments, actin and myosin, to bind together. And that binding is what creates movement. Depending on what you're doing, your muscles might do an isometric contraction where the length stays the same, a concentric contraction where the muscle shortens, or an eccentric contraction where the muscle lengthens out. Of course, muscles kind of need a framework to pull against, right? Let's do a really quick, rapid-fire look at the musculoskeletal system. Bones don't just give you structure, they actively store vital minerals. And tucked right inside them is your bone marrow, which is basically a 24/7 factory pumping out blood cells. Then we have the genuinely incredible design of our joints. Hinged joints give you that swinging motion in your elbow and knee. Gliding joints allow for limited movement by letting smooth surfaces slip over one another. And the ball and socket joint? That gives you unparalleled freedom, letting you move in practically every direction. Now, the ultimate biological goal of all these surviving systems is, of course, reproduction. Looking at the human reproductive system, we see some highly specialized structural roles. In males, the testes produce sperm and testosterone, while the seminal vesicles secrete fructose, just to give that sperm the energy it needs. In females, delicate little structures like the fimbria sweep the ovum forward. The whole female reproductive system relies on a remarkably precise menstrual cycle: menstruation, followed by the follicular phase, then ovulation, and finally the luteal phase. And this entire timeline is strictly governed by hormones, with luteinizing hormone promoting ovulation and progesterone stepping up to maintain the endometrium during that final phase. All right, on to Section 3, Animal Behavior and Conditioning. We're looking at stimulus and response. Behavior starts with what's already built in. By definition, innate behavior is genetic, it's an instinctual response specific to a species, and crucially, it does not require any learning at all. These are the survival tools you are quite literally born with. Think about spiders just instinctively knowing exactly how to weave these intricate webs to catch prey, or the sudden reflexive jerk of your hand the second you touch a hot stove. Even a cockroach scurrying away from a light, which is a behavior called negative phototaxis, is an entirely innate genetic instinct. But what about the behaviors we pick up over time? Let's contrast two foundational learning methods here. First is classical conditioning. This one is all about association, linking a completely neutral stimulus with an unconditioned natural stimulus. Then we have operant conditioning. This is a totally different ballgame. It relies heavily on the consequences of an action, using positive reinforcements or punishments to actually shape a specific behavior. To make sure this concept sticks for good, just remember this easy rule. If a dog starts salivating just at the sight of food, or maybe hearing a dinner bell ring, that is classical conditioning. The dog associated the bell with its natural instinct to eat. But if a dog purposely sits on command because it knows it's going to get a treat for doing it, that is operant conditioning. The dog learned that taking a specific action results in a very positive reward. And that brings us to our final grandest leap. Section 4, Population Ecology and Conservation. Populations and the planet. To understand massive populations, ecologists essentially play a giant game of tag using the mark recapture method. Used mostly for mobile organisms, you capture a sample group, mark them, release them back into the wild, and then recapture a new sample later to estimate the total population size. Now, if you're studying sessile or non-mobile organisms, like a big field of plants, you wouldn't do this. Instead, you'd use a technique called quadrat sampling to estimate their numbers. Once we know how many organisms there are, we can track how they multiply. First, you have exponential growth. This creates a really steep J-shaped curve because it assumes growth happens completely without any limits. But we all know the real world has limits, right? So that brings us to logistic growth, which creates an S-shaped curve. Why the difference? Because density-dependent factors, things like food scarcity or disease, start to increase as the population grows. This eventually forces that limitless J-curve to slow down and flatline right at the environment's carrying capacity. So how do these populations actually look out there in the wild? Well, demographics show us how organisms are spatially distributed. You might see uniform dispersion, where animals are spaced out perfectly evenly. You could see random dispersion, which is the absolute least predictable setup. Or you might see clumped dispersion, where everyone gathers in tight clusters. Demographics also let us track lifespans. For example, humans exhibit a type 1 survivorship curve. That basically means we are characterized by extremely high survival rates throughout our early and middle life. But as we know, our high survivorship definitely comes at a serious cost to the environment, especially as rapid industrialization depletes our natural resources. Take bioaccumulation, for example. It's a process where harmful toxins build up in organisms, and terrifyingly, these toxins actually multiply in concentration as they travel higher and higher up the food chain. This is exactly why modern conservation efforts, things like establishing botanical gardens, prioritizing renewable resource management, and fiercely protecting habitats, are absolutely vital for the health of our planet. Which brings us to our final and maybe most provocative thought for today. As our own exponential human growth continues to put immense strain on our renewable resources, how are we going to use our biological blueprints, our genetics, our learned adaptations, our intellect, to adapt and survive the carrying capacity of Earth? It is a massive question, one that connects those tiny microscopic blueprints we started with directly to our macroscopic survival. Thank you so much for joining me on this incredible crash course today. Stay curious, keep learning, and I'll see you next time.